Hello everyone, I'm Mr. Furlong, and today we're talking non-Mendelian genetics. In the last video, we looked at doing Punnett squares and how when you're doing something that follows Mendelian laws, works really well. The problem is there's a lot of traits that don't follow Mendel's laws. And so you can't use Punnett squares to predict them. So let's take a look at a couple of reasons why. One is that they can be linked genes. Linked genes are genes that are located close together on the same chromosome. Now technically, any genes located on the same chromosome are going to be considered linked. The closer they are to each other, the more linked they are. And the closer they are, the less likely they are going to separate during meiosis in the process of crossing over. So for instance, we know that in crossing over, at any point along this chromosome, it can break off. The only time we could use a Punnett square is if crossing over took place right between, say, gene 1 and gene 2. That's not very likely to happen. Another example are sex-linked traits. These are genes that are located on the X chromosome, which technically would also be a linked gene. One thing to remember is that the genes on the X chromosome, they're not the same genes that are found on the Y chromosome. And really, the X chromosome doesn't really have anything to do with gender. That's going to be the Y chromosome that determines that. Specifically, there's going to be the SRY gene that determines the sex of the individual, not necessarily the chromosome Y. Now, the probability that linked genes will segregate as a unit or segregate together can be used to calculate the map distance between them. So the map distance tells us how close together a pair of linked genes actually is to one another. For example, if the genes separate from each other during the crossing over about 5% of the time, we simply say that the genes are 5 map units apart. If they separate 50% of the time, they are 50 map units apart. So think about what that means. The more often they separate during crossing over, the farther apart they are on the chromosome. The more times that they stay together after crossing over, the closer they are together, because the only time they're going to separate is if crossing over takes place between them. And if they're real close together, they're not going to separate very often. We spend a lot of time talking about these single allele traits. However, many traits are the product of multiple genes acting in varying combinations. And these traits don't segregate via Mendelian patterns. Let me give you an example of this. Human height. This is a trait that has multiple genes. So you have 6 alleles for height. And some are going to be dominant, some are going to be recessive. So I'm just going to use 3 letters here, A, B, and C. And let's say we have 2 people who each are of average height. So they have half of their alleles are dominant, half are recessive for height. They could have children that could be really tall or really short, or it could be average height like their parents are, right? So each parent is going to donate 3 alleles. Which alleles that they donate in meiosis is going to be totally by chance. It could be that one child gets all the recessive alleles. And if they do, they're going to be shorter than the average person. If a child gets all the dominant alleles, they're going to be taller than the average person. But more likely, they're going to get some combination of dominant and recessives. And they're going to be a person of average height. If you look on the graph, we can see that very few people are going to be really short. Very few people are actually really tall. Most of us fall somewhere in the average category. Now, what's average? It depends on if you're male or female. So here's the thing. Not all of our genes are on a chromosome in the nucleus. Remember, we have DNA somewhere else in the cell. We have it in our mitochondria. So some traits result from non-nuclear inheritance. Besides mitochondria, chloroplasts also have their own set of DNA. The chloroplasts and mitochondria are going to be randomly assorted into the gametes and the daughter cells during cell division, during meiosis. But let's focus on animals. In animals, the mitochondria are going to be transmitted by the egg and not the sperm cells. So all the sperm cells are going to give the offspring is just going to be their chromosomes. Everything else is coming from the egg. All of your mitochondria that you have is going to be genetically identical to your mother's mitochondria. So all of these traits are going to come from mom, not from dad. So how would we be able to determine this if we were, say, looking at a pedigree? Well, first off, notice in generation 1, mom has this gene is found in the mitochondria. Notice she passes it on to each one of her children. Now, could this have been a trait that was found on a numbered chromosome and she passed it on to all of her children? Certainly it could have. However, if you look at generation 3, notice all the grandchildren that have it, they all have a mother who was one of the daughters of this person. So we can follow that trait from mom going to all of her children, but then only her daughter's children would also have that trait. It doesn't matter what dad's genes are. You can see the 3 boys that she had, none of the grandchildren had that, because even though it's in their mitochondria, fathers don't pass on mitochondria to their children. So that is non-Mendelian genetics, which, as a reminder, tells us that we can't use Punnett squares for all types of inheritance. I'll see you in class.